So I'm going to be talking about this type of diversity on the wings of butterflies. These are satyrid butterflies. There's a lot of eyespots that this family of butterflies uses. And I've been interested for a long time in what creates this type of number diversity on the wings of these insects. My talk actually has these three parts, so I'll be talking a little bit about eyespot origins and diversification in terms of number, and then the ecological function of eyespots, addressing why we have different numbers of eyespots, primarily on forewings and hindwings, and then a little bit about the genetic basis of those differences. So work by Jeff Oliver and other people in my lab a while ago by scoring using museum specimens, and this is 400 genera of nymphalid butterflies. By scoring presence or absence at the tips, we sort of inferred that eyespots evolved once, so they are homologous across all these species. It keeps varying with the new phylogenies that come out, but nowadays it's about 75 million years ago. And they evolved in the sister lineage to the Danainae, which are butterflies that look like this, that have simple spots on their wing and other things, but they don't have these concentric rings of color. So when we looked at a variety of genes, these are developmental genes, and we looked at 5 of them: notch, distalis, engrailed, spalt, and antennapedia. And we looked, and here I'm combining data also from Patricia Beldade's lab. When we looked at the 5 genes in a variety of species with eyespots, what we found is that 4 of them, but not antennapedia, were reconstructed. So because, for instance, this yellow gene, the orange gene over here is distalis, most species with eyespots have distalis. And so we reconstructed the expression of the gene in the center of the eyespots to the same node here when eyespots evolved. The same thing for spalt. So this is the red gene over here. It's expressed in all these species. And that means that we reconstructed over to the same node. And then these other 2, engrailed and notch, are a bit more patchy. So many species don't carry these genes. They still have eyespots, which means that these genes might have no function in eyespot development whatsoever. But the reconstruction of the origin of the expression of that gene also falls to the same exact node when eyespots evolved. Antennapedia is the exception. There is a biblidini over here that has antennapedia, and then the saturnine clade, the ones that have a lot of eyespots on their wings, also have antennapedia. But there seems to be an independent recruitment of this gene in these 2 separate lineages. These species have eyespots without antennapedia there, so they don't need it. Okay, so what do we conclude from this? We conclude that potentially the origin of eyespots at this place in the phylogeny is connected to the recruitment of a slew of genes together potentially into this pattern that we see here in the center of the wing pattern, the eyespot patterns. So we suspected that there was a gene network co-option event, not just the co-option of 1 gene, 2 genes, 3 genes, but potentially a whole network of genes that got recruited at this moment in time when the trait evolved. So to try and figure out what network might have been recruited, Surya Murugesan in the lab, he dissected a bunch of tissues in the pupil of a Bicyclus aninana, including the eye, the antenna, the T1 legs, the wing margin, the whole wing, and then little squares of tissue containing either the eye spot or other bits of control tissue. And what he found was that eye spots were clustering in terms of their transcriptome map. They were clustering with antenna. So this was kind of interesting. They were not clustering with legs over here. They were not clustering with the whole wing, in fact. So the whole wing was clustering outside. They were very similar with antenna. So if eyespots and antenna and potentially other limb-like structures, if they share the same network, what we expect is that if there is some sort of combinatorial group of genes in some sort of temporal order of expression, this gene activates this gene by binding to its cis elements, cis regulatory elements over here, then this one activates these other ones and so forth. So if we have a shared network between eyespots and antenna, we expect to see the same type of regulatory interactions between the genes. So some genes being upstream, others in the middle, others downstream. In addition, if eyespots and antenna share the same network, then genes that are in the middle of the network, they should be using the same cis elements to drive these genes in the middle of networks, both in the context of the antenna or the leg in the head, and the same cis element is expected to drive the gene again in the novel context, which is in the eyespots. So the idea here is that if we find these cis elements, if we knock them out, we should see two traits or more being affected if we have pleiotropic cis elements that represent co-opted networks. Or in addition, if we take these cis elements from genes in the middle and we attach them to GFP, we should see GFP expression in the ancestral context and also in the novel context, which would be the eyespots. So to identify these cis elements, by the way, we did look at these regulatory interactions, but I won't have time today, so I'm skipping that part. I'm going straight to the cis element sort of test. And we dissected all these tissues that I've already shown before, the eyespots, the legs, the wings, etc. And we focused on areas of open chromatin that were open in antenna, potentially in other limbs, and also in the eyespot dissected region. The eyespots is the black area here. And so for distillus, which is a gene that has all these exons over here, we found these three peaks that were not containing transposable elements, because most of the other peaks around here contain transposable elements and they're open peaks. But we found these three peaks, and then we focused on one of them, this distillus 319, a TAC-seq peak, and we targeted it with guide RNAs, and we tried to delete it. So the idea here is, if eyespots co-opted some type of pre-existing network that is involved in building antenna, we should be able to see defects both in the antenna and in the eyespots if this ends up being an eyespot Cree or an antenna Cree or something like that. And that's indeed what we found. So by deleting that cis element, we got disruptions in the antenna, and we also got disruptions in the eyespots. We also had problems with pigmentation and so forth. A variety of different phenotypes. Legs also got truncated. Some individuals didn't have wings at all. We did the same exact experiment, but with a different gene in the network. So we tried Spalt, which is downstream of distillus. I'm not showing you this data, but we did the exact same thing. And we targeted the gene Spalt, which also potentially has a pleiotropic Cree. And so when we did that with Spalt, we also got effects in multiple traits. Some of them were the horns in the head. Antenna got pigmentation defects, and eyespots also went missing. So the final thing we did was we took the distillus Cree and we attached it to GFP, and we produced a transgenic line of butterflies. And what we observed was that that specific distillus Cree was driving gene expression in the antenna, in the mouth parts, in the thoracic legs, and also in the eyespot centers in the larvae of these butterflies later in development. And so, again, this is very strong evidence that there has been this network co-option going on, that we have a network that was ancestrally involved in producing these outgrowths, antennae and also legs and potentially even wings. This network was driven by some sort of head gene in this case, but it involved distalis and spalt. It involved this specific cri of distalis, this specific cri of spalt, and this little group of regulatory elements and their associated genes got co-opted as part of a co-opted network and got redeployed in evolution to produce these eyespot patterns. So eyespots share the same genes, the same interactions, which I have not shown you, and the same cris with limbs. Now, going back to this original question, when we have variation in eyespot number on the wing, what exactly is going on? So I've just shown you that we've mapped eyespots to about 75 million years ago, but when they originated on the wing, where did they originate as a one unit or a couple of units on the wing? Or did they originate across all of the wing sectors that you see here from this species from Cambodia, and then got slowly repressed by different alleles to create the type of variation that some species now display. So what is the original number of eyespots when eyespots appeared on the wing? To solve this, to answer that question, Jeff Oliver again did the same type of ancestral reconstruction analysis. And what he did was, now instead of just scoring at the tips of the tree, the same number of 400 tips that we had before, instead of scoring just presence or absence of an eyespot, he scored presence or absence for an eyespot in every single sector of a butterfly wing. So there was massive scoring going on, dorsal surface, ventral surface, everything. And what he found is that by using hypothesis testing in phylogenetics, he asked the question, is it, this eyespot is the most common eyespot, it's one of the oldest eyespots, but he asked, is this the most likely ancestral state at this place where eyespots evolved, again, as the sister lineage to the Danainae? So is this the most probable ancestral state, or is we call it the nymphallid ground plan, the kind of stereotypic, it's an hypothesis of all the possible pattern elements that butterflies carry on their wings. So is this the most likely ancestral state, or is this the most likely ancestral state, or something intermediate like this? Are these the most likely ancestral states? And what he found was that either 4 eyespots on the ventral hindwing or 5 eyespots on the ventral hindwing were the most likely ancestral states for butterflies. And some lineages of butterflies still to this day only have eyespots on the hindwing ventral surfaces and have never yet diversified into having eyespots either on the forewing or on the dorsal surfaces. So it's still, this type of origin of eyespots restricted to the hindwing is still something that has a signature in the diversity that we see in butterflies today. So we were interested in understanding, why is it that eyespots evolved restricted to the hindwing? And when we think about hindwings, we think about Hox genes, and in particular, one Hox gene that is hindwing specific, which is called ultrabithorax. So we wanted to know if eyespot origins might have something at all to do with the presence of this Hox gene called ultrabithorax. Ultrabithorax is expressed just in hindwings in pretty homogeneous domain across the whole cells, but is expressed a little bit in higher levels in the eyespot centers in the larval stage. So what we did to test the role of ultrabithorax in eyespot development was to knock it out. This was done by Yuji Matsuoka in the lab. And what we observed was what we typically observe with UBX transformations, which is you get forewing identity being present in the hindwing. So this wing, it's a mosaic, just like Arnaud has shown you. We are looking at a mosaic individual where not all cells are UBX negative. But what we see already is that in this sector here, which is amplified here, called the M3 sector, which traditionally has an eyespot over here, when you remove UBX, the eyespot is gone. So this tells you that UBX is actually an essential gene for eyespot development in this sector over here. In this other sector, something else is happening. So UBX is, in this case, seems to be repressing the size of this eyespot because the wild type size is about this big. But if you remove UBX, you get a much bigger eyespot that looks like the forewing eyespot. And so there are these different functions of UBX relative to eyespot development in different sectors of a butterfly wing. But this experiment kind of suggested that perhaps UBX in primitive species within the butterflies, if we were to look at additional species and do this type of experiment in basal lineages, it might be the case that early on UBX might have been solely an activator of the eyespot network. We don't know. We also looked at a separate Hox gene that Patricia's lab had identified as being an important gene connected to eyespot development, and that is antennapedia. That's a Hox gene that is, in most insects, uniformly expressed in low levels. In fact, it had been suggested that antennapedia had no role at all to play in wing development, but that is not true. Antennapedia is actually quite important for wing development in a variety of different species. But in the butterflies with eye spots and in specific lineages, remember antennapedia was one of those late genes to get added to the network. It was not essential for eye spot development, at least in Bicyclus aninana. When we knock it out in forewings, we do get eye spots being removed altogether. But when we knock it out in hindwings, we only see a reduction in eye spot size and the elimination of the white centers of the eye spots. So it has two different roles in forewing and hindwing relative to eye spot development. It seems to be essential for eye spots in forewing, but just a gene that makes eye spots bigger in the hindwing. And so this is interesting, and we think that this non-essential role in hindwing might be because it is there expressed also with UBX. And so UBX and antennapedia might have similar roles in hindwing. And so if you remove antennapedia in the hindwings, the eye spots suffer a little, they become a bit smaller, but they don't disappear altogether. But when antennapedia got recruited to the eye spot centers of both forewing and hindwing, it's the only Hox gene that is present in forewings. And thereby, it might be taking over the activating effect of UBX, but now in the forewing. So it is an essential gene in forewing eye spots, but not so essential in hindwings. So we think that when antennapedia got recruited independently, in this lineage, the Biblidinae, and here in the Satyrini, this was one of the instances in evolution when we were able to finally get eye spots colonizing the front wing, when before they originated solely restricted to the hindwing. So the Hox genes might have been super important for initially the origin of eye spots altogether, although, as I just mentioned, we only have data from one species in the Satyrids. But then antennapedia helped out carry over the network and activate the network in the forewing afterwards. So we might have had species without eye spots. UBX was potentially an activating gene to lead to the situation. The bringing on of antennapedia to the network itself, to the center areas, allowed eye spots to move over here. And then there's been a lot of other diversification in terms of eyespot numbers. And so this is what I'm just going to talk to you a little bit about. When we just scan different species and we count the number of eyespots that are present on forewings versus hindwings, there's always a big difference in the number. There's always many more eyespots on hindwings than you find on forewings. So this is very consistent and it's very prevalent across the nymphalids. So why do we have this bias? Why do we have this difference in the numbers between forewings and hindwings? So we did some predation experiments. Several people in the lab worked on this. We used a mantid predator, and we realized that mantids are actually quite distracted by these pattern elements on the edges of the wings. And if we train a mantid to go up a ramp, and you put a butterfly with a piece of banana in the middle here, the mantid will go up and then slowly climb up the ramp, and then when it's very, very close to the butterfly, it strikes its raptorial legs, strikes the butterfly wing. And when we do this experiment, with two different seasonal forms of this Bicyclus aninana butterfly, one that has prominent eye spots and one that has very reduced eye spots, we observe completely different locations of the attack of the mantid. So the mantids primarily attack the hindwings in the form that has the prominent eye spots, but they primarily attack the head and the thorax of this, the form that has very reduced eye spots. So having eye spots in general is a good thing, because the butterfly can survive even though it is attacked. The attack is initiated earlier because they detect the butterfly and they detect the eye spots faster. But while they attack the butterfly, because they focus on this part of the body rather than the head and the thorax, then the survival of the butterflies with eye spots is increased, and they lay more eggs and so forth. So when we were trying to understand this idea, why is it that we have more eye spots in general on hindwings and then fewer on forewings, we made use of this spontaneous mutation in our lab colony called Spotty. And Spotty has 2 extra eye spots on the forewing. And so we use these 2 forms of the butterfly, both wet season forms, both with prominent eye spots, again in experiments with mantids. And the idea was the following. If we have more forewing eye spots, perhaps they attract even more attention from these predators. But if attacks on the forewing are somewhat more detrimental to the flight escape behavior of the butterfly, because the forewings are very important takeoff wings, then we might have an impact overall, and there will be a selective advantage of having fewer eye spots on the forewing. And so that's what we tested with these experiments, and what we found was that, in fact, there was such a thing. There was the spotty individuals had a much faster decline, so they went extinct in these cages much faster than the wild-type individuals. This is the heat map of where all the damage by the raptorial legs was done on the wings. And you can see that in spotty individuals, the center of most of the attacks shifted forward on the wing. So the attacks on spotty wings were more like both wings got attacked, but the attacks on wild-type individuals with the more visible patterns being more lower down, the center of mass of the attacks was more towards the hindwing. The center of mass of the attacks for the spotty individuals were more shifted forward, and there was, as a consequence, differences in fitness. So eye spots are good for you, but you have to watch out where you put them. You don't want to put the eye spots on the very important flight takeoff wing, which is the forewing. And so, just very briefly, how long do I have? 2 minutes? 1 minute. So just very briefly, we tried to figure out what is the genetic what is this gene mutant, what is spotty and what is a different one called missing that has the exact opposite effect of spotty. It removes 2 eye spots from the same sectors that spotty adds eye spots to. And so a QTL mapping analysis mapped spotty to this chromosome 10, to this particular interval. These are the people that worked on that project. 2 different recombinant breakpoints narrowed down that interval to about 70 kb. And there's this gene called outspread in this area. There's other genes as well. Outspread seems to be the only gene that is differentially expressed between spotty and wild type. The other genes are not so much. And outspread, this is not the grade we're working now on the expression, but outspread seems to be a sector-specific gene. So it's kind of like a Hox gene, but at smaller levels. So it's expressed at higher levels in these sectors, but then in these sectors where the spotty mutant is present, there's lower levels of outspread, and then outspread is expressed again at higher levels at the edges. The missing mutation is also mapped to the exact same interval, chromosome 10, and here you have the opposite. You have 2 eye spots missing on the wing, and here the breakpoints are in between the same area where spotty is located. So the breakpoints for missing are a bit wider, so this is a 4.5 MB interval. The spotty interval is within that interval, so both of them are mapping to the exact same region of the genome. So we're now doing CRISPRs with outspread to try and find out if that's a good candidate or not to regulate eye spot number in a finer way than what these Hox genes have been doing. So just in summary, butterfly eye spots are novel traits that evolved first on hindwings, then on forewings, later in evolution. Eye spots attract predator attacks, but too many eye spots on forewings reduce fitness. So this may explain why fewer eye spots are found on the forewings in general in nymphalids. Eye spots appear to originate from the co-option of this ancient appendage GRN. They seem to be flat limbs redeployed on the wings. The origin of eye spots in hindwings might have depended on UBX, but additional comparative work is necessary. And UBX might be essential for eye spot development on some eye spots. We know it is essential in some eye spots in Aninana. And then the appearance of eye spots on forewings might have later been due to the co-option of the second Hox gene that might have allowed the UBX function to take over the role in the forewing. And genes like spotty and missing are further allowing individual control of specific eye spot number diversity and adding them on forewings and removing them on hindwings. And the genetic locus of these 2 more detailed genes seem to be the same, approximately the same, but we're still working on the identity of this gene. This is a lot of work from very different people over many years, and so I thank them all, and thank you for your attention.